In developing new clinically relevant drugs, we often look towards altering the behavior of various proteins to alter a biochemical process. In targeting some of the many proteins involved in DNA replication, we often see a therapeutic application in treating cancer. However, there are significant applications beyond chemotherapies, as preventing cell reproduction and viability in bacterial cells, for example, can lead to some pretty novel treatment applications. What if I told you that some painkillers can actually target bacterial cellular machinery, leading to an unexpected antibiotic effect? At the site of DNA replication, a multi-protein complex is assembled, each part of which is highly important in promoting efficient DNA replication. While DNA polymerase often steals the spotlight in that it's the protein responsible for assembling new DNA, it has its limitations and needs help from other proteins to do its job. For one, DNA polymerase is not actually able to open up and get into double-stranded DNA. So another protein, DNA helicase, must help it by actually unwinding the DNA. But even at this point, DNA polymerase can't start making new DNA until another protein, DNA primase, makes almost a scaffold so that DNA polymerase has somewhere to start. There's also what is known as a single-stranded DNA binding protein that stabilizes the unwound DNA while DNA polymerase does its job. But even after all this help, there is yet another protein that is essential to DNA synthesis process, the sliding clamp. This protein is comprised of 3 identical subunits and forms a sort of donut shape, which after help from yet another helper protein, the clamp loader protein, encircles DNA and becomes associated with DNA polymerase. Now, here's why this protein is so important. It really strongly promotes the processivity of DNA polymerase. In other words, polymerase activity is stimulated and the nucleic acid products that the polymerase makes are longer than the many small fragments that would be made if not for the sliding clamp. Thus, this protein is crucial for cell viability and replication. So, looking back to drug development, now that we know what the sliding clamp is, we can find a molecule that can bind to this protein to alter its activity. For inhibition of the beta clamp, which is a sliding clamp subtype found in bacteria, a drug molecule must bind tightly to prevent its action. Various molecules have been found to be able to do this, including synthetic peptides, small molecules such as a dibrominated aromatic ring called RU7, as well as natural products such as a substance called grisilamycin, which was derived from Streptomyces bacteria. All of these examples demonstrate potential for antibiotic therapies against organisms that use the beta clamp, which can allow for treatments such as antibacterials against mycobacteria, for example, with the aforementioned grisilamycin. Interestingly enough, what was found in 2014 is that some drugs in a class of commonly used painkillers are inhibitors of the beta clamp as well. This class of drug is called NSAIDs and actually includes everyday medicines such as Advil and aspirin. It was found that the NSAIDs carprofen, bromfenac, and vitoprofen directly inhibit growth of E. coli and other common bacteria. Again, this occurs because bacterial cells are not able to replicate their DNA as efficiently, if at all, leading to decreased cell replication and viability. How this inhibition actually works is rather complex, but in short, it was found that those NSAIDs bind to one of 2 subsites of the beta clamp dimerized structure, inserting a hydrophobic moiety deep within, while leaving their aromatic ring groups on the protein's shallower region. All 3 of these drugs have carboxylic acid functional groups, allowing for hydrogen bond formation and potential stabilization. While this research is still relatively new and more experimentation must be done to fully understand this inhibition mechanism, this opens up future possibilities for new synthetic antibacterial drugs. This is groundbreaking because we're constantly in the search for new antibiotics as we battle against the inevitable evolution of antibiotic-resistant bacterial strains. We're running out of antibiotics and significant research needs to be dedicated to ensuring that we are able to keep up with bacterial evolution. We need to recreate the prolific development of antibiotics in the mid-20th century that allowed for the production of all the 20 classes of antibiotics we've been using for the past 60 years. So, the next time you take an over-the-counter painkiller such as Advil, remember that it's drugs similar to these that may be key in finding new and more effective antibiotics that may be used in the future.